in this amazing class in front of me is APBio 7th period, and we are reviewing Unit 1, not only for our semester final, but because we're also thinking about what? AP exam. Excellent. All right, so talking about this unit, here we go. Read through this. There are no highly suggested reading and thinking. I don't put that on the final. Okay, so just take that off your plate right now. Does that feel good? Okay, so read through these. Maybe identify things that make you nervous so you can go back later and review those. Just going to give you about a minute to read through it. This was, remember when I practically told you all 4 essay questions? Yeah, that's not happening. That was from when we first did it. Okay, you good? Did you highlight? Still looking? Still not? Oh, it's not loading for you yet?
It's been very slow. Hey.
Okay, guys, let's keep going. Look at how many slides I have in this. 64. Okay, we don't have 64 minutes, right? We have half that. That's like 30 seconds a slide. So can we, you're going to have this, right? Will I put it in Google Classroom like I always do? Okay, so you will have it. All right, so let's move on if we can. Come on, baby. Come on, baby. Here we go. Characteristics of life. And we're going to do organization. Living things are organized, made of cells. They acquire materials and energy. They maintain homeostasis. They respond to stimuli and reproduce. They grow and develop, and they adapt and die. And when we say living, do that part with me. Living things are organized, made of cells. Let's talk about how they're organized. Atoms, molecules, organelles. Do all cells have organelles? Which ones do? Okay, go again. Atoms, molecules, organelles, cells. Why are we doing this? Life starts good. Tissues, organs, organ systems, organisms, population, community, ecosystem, biosphere. We're good? See? That could just be the final. You'd be great. Yes. All right. So, pause. That is life, and that is how life is organized. In APBio, these are your 4 big ideas. Look what number 1 is all about. What is number 1 about? Evolution, change over time, right? So that's a big theme. Number 2 is all about utilizing what? Energy and molecular building blocks to grow, reproduce, maintain stasis. Number 3, living systems retrieve, transmit, and respond to what? Information. Remember when we just finished that unit about cell communication, how you can talk one cell to another? So that's a lot about that. And then big idea 4, biological what? Systems. Populations is just one species. Population. Community is what? Multiple species. And that what, right? And then ecosystem, what are you bringing in? Environment. And then biosphere is where all living things exist, right? All right. Okay, that's only a little scary. Okay, now we talk about the first big idea is evolution, right? And then everything now, when you see this, you should be able to recognize some things on here that you didn't recognize when we first learned this, right? If you look over here, I can see alanine. These are amino acids joined to form a what? Protein, right? What do I see right here? What's this? I see a nucleotide right there. Do you see that nucleotide? And then here, what's that? That's glucose. That's a carbohydrate, and that's built in a chloroplast. You know that. What's this right here? Fatty acid. Do these look like they're saturated or unsaturated? Saturated. Solid or liquid at room temperature? Solid. Plant or animal? Animal. Boom. See how smart you are? And then life shares the same molecules and reactions, points to a common ancestor. Okay, I don't know if you can see it, but this says glucose, and then I think I see some G3P in here. What reaction starts with glucose? Cellular respiration, what? What's the first step? Glycolysis. Who does it? All cells, right? So this is showing that we have these same important organic molecules and these same biochemical reactions that we probably have a common. See how smart you are? So smart. Okay, now there are many things that can cause evolution. We haven't gone into that yet, right? We haven't really talked about that very much. We haven't hit that unit yet. But natural selection is one of 5 things. What do you hear in natural selection? Nature is selecting who has the best traits. We've started to talk about that, right? We talked about alleles. And whoever has the best traits is the one that's going to survive and make more of those traits in their offspring. And a measure of that is called what? Starts with an F. Fitness, right? Fitness, right? Is whoever can pass on their genes. And all evolution is who is best fit for that particular environment. Whoever's best fit gets to live, have more sex, and have more babies. They will survive and reproduce over time, and they will become adapted to that particular environment. Do you agree with that? That's all evolution is. All right, that's that part. Are we good? Can I keep going? Okay. Then, okay, we talked about this. We talked about how nutrients cycle. Remember that? Nutrients cycle, but energy flows. We also know energy is neither created nor destroyed. It just transforms. And every time it transforms, you lose some as what? Heat. And we can see that right here, can't we? But the carbon will be conserved. Carbon is just moving from carbon dioxide. It's getting reduced into glucose or G3P. And then that glucose is used by what cells? All cells to do cellular respiration and take that chemical energy of glucose. And you're going to make ATP out of it, yes? But along the process, do we always get all the energy out of it? No. What's the way we can maximize cellular respiration? The use of oxygen. And what is that called? Aerobic respiration. And how much ATP do we get for every glucose molecule in aerobic respiration? 32 to 30. Yeah, if we don't include the second law of thermodynamics, it's 36 to 38, but we know it's less than that, right? But how many do we get if we just do glycolysis followed by fermentation? 2, right? So we can maximize our output with the presence of oxygen. And what is that called? Oxidative phosphorylation. There you go. See how smart you are? Okay, so that's that part. Now, genetics. We're starting to get into genetics, right? And actually the next unit will break down the DNA, but we know that our chromosomes have genes on them, and we know alternative forms of genes are called what? We just learned this recently. It starts with an A. Alleles, right? And that codes for our traits. If you change the DNA, will you change the traits? Yeah. How does that information get out? It's in the DNA, and then I need to make a copy of it. What's that copy that I use? What kind of RNA? What does M stand for? Messenger RNA. And then it goes out and it gets on the workbench. What is the workbench? Ribosome. And that mRNA will then get translated into a protein. Okay, that's all we're talking about here. That's easy. Okay. All right. Did we already do this? Yeah, so I'm just moving on. Okay, emergent properties, right? Can a tissue do more than a cell? Yes. Can an organ do more than a tissue? Can an organism do more than a kidney? Yes. Okay, that's just emergent properties. As you go up in evolution, you acquire new properties. All right, easy peasy. Next. All right, cooperation and competition. We're going to get more into this. Remember, this was the overview of the year. But you have populations. This is a population of what? Ants. They are working cooperatively together to get to this next leaf. That's what it's talking about. And when you look at things like that, you're looking at behavior, how an organism responds to their environment. All right, next. Oh, scientific method. Meh, 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 meh. Keep going. Da, 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 da. Okay. All right. So we make observations. We know that. All right. We observe what's going on, and then we ask a question. I wonder how light affects photosynthesis, right? I wonder how the amount of carbon dioxide affects photosynthesis. Yes? Okay. Then we're going to form a hypothesis. If I provide carbon dioxide to a plant, then it will do more photosynthesis, generating more oxygen, which will make my little leaf disc float. Right? That would be a hypothesis. If, then, because. What kind of hypothesis is that? Research. Okay. Or I could write a null hypothesis, which says what? Yeah, the amount of carbon dioxide does not impact photosynthesis. Follow? Okay. Either way, if it's a research hypothesis, I'm probably trying to support that idea, right? I'm going to collect my data and I'm going to look at the difference between what I observed and what I expected or hoped for. And I'm hoping that it's a very small difference and my chi-square number is very small and I can accept my hypothesis. Did you follow that? But if it's a null hypothesis, I don't really believe it's null, do I? No, I'm just trying to make some sort of connection by saying there is no connection. I'm saying carbon dioxide has no impact on photosynthesis. Then I'm collecting my data and see if I disprove it. To show that I disprove it, I don't want a little difference. I want a large. Yes. Then I can reject my null hypothesis. And if I reject my null, I'm actually supporting my alternative, my research hypothesis that CO2 does have an impact. Did you put it all together now? Okay. All right. I've got to analyze my data. Might need to do a little math to analyze my data. And then I'm going to do represent my data, let's say in a graph. Remember, your independent variable goes on your x-axis. That's the one that you are in charge of, right? All the other variables are controlled. Like you use the same amount of water, the same size beaker, the same size leaf disc, right? Your independent variable was the amount of carbon dioxide in the water, right? I'm just picking one lab that you're familiar with, yeah? So that's my independent variable. My dependent variable, I believe we graphed ET50 if we graphed it all, right? When half the disc floated, right? So that is on my y-axis. Does that make sense? Independent, what I choose is on the bottom. I'm like laying it out. I choose this. Then my data is on the y, yeah? Okay. Variables, we already talked about this. Independent variable I choose, another name for that is experimental variable. And then your dependent variable is your data. Constants, you're keeping everything the same. I think you're comfortable with that, but you can look back over this in this presentation and what controls are, right? Basis of comparison, quantitative versus qualitative. Which one can I grab? Quantitative, right? Qualitative is it's greener. It looked better, okay? We already know the difference between a research and a null, right? A research hypothesis says there's a connection. The amount of CO2 does affect how much photosynthesis. A null hypothesis in H0 says there is no connection. Kind of already went over that. Oh, we know this chi-squared, right? That's easy peasy. Observed minus expected. What did I see from what I got? I square that difference. How big is my difference? Put it over what I expected. And I do that for every different data point that I'm looking at, right? Every different observation that I'm making. Now, let's say I make 5 observations. That's going to be important because I have 5 observations, but when I go to this chart right here, I have to be able to look it up by my degrees of freedom, yes? So if I have 5 outcomes, how many degrees of freedom do I have? 4, okay? And if it's 4, I can take all the way up to here, right? I can take up to here, and then after that, then this is the reject zone right here. Okay, I'm going to reject if it's 9.49 or a bigger number because it's so off. My chi-square value is so big, it tells me to reject. Anything here, I can what? Accept. Okay? All right. That was chapter 1. Yay. Chapter 2. It's okay. There's only 3 chapters here. So do this with me. The atom story. Quickly. Atoms are made of protons, neutrons, and electrons. The protons and the neutrons are in the nucleus, atomic mass. The electrons are in energy levels 2, 8, 1832. 8, 8, 8 is great, octet rule. Okay, there's a lot in that, right? If I'm talking about atomic number, that's the number of what? Protons. That tells me what element I have, yes? My atomic mass is protons plus neutrons. In every element, is its neutron number the same? No. Has everybody signed in on the sign-in sheet? Where does it need to go? Raise your hand if you need it. Wow, how did that happen? Okay, why don't we start it right here? Yeah. Okay. What was I talking about? Varying the number of neutron numbers, right? What do you call it when you vary the number of neutrons? Isotopes. Good. Can I vary the number of protons for an element? No, that changes it to a new element, right? Can I vary the number of electrons? Yes. What is that called? And if I lose electrons, I will have a what kind of charge? If I lose electrons, positive. And if I gain electrons, I'll have a. And what do we know about opposites? They attract, right? And that what kind of bond is that? Ionic bond. We know elements want to get happy. There's two ways to make an element happy. You can either what? Fill up outermost shell, or you get it to 8. 8 is great. What's that called? The octet rule, right? The reason why one atom might bond with another atom is to get happy, right? So they can do it one of two ways. One way is to share, and when you share in pairs, a pair of electrons. If you share one pair, it is a single bond. If you share two pairs, double. So, right? We know that. Okay? And so when you share for hydrogen, hydrogen is only one proton and one electron, yes? Okay? So its outermost energy level will hold how many electrons total? How many? 2, 8, 1832. So for hydrogen, who has one electron, it can only hold how many? 2. So it has one electron. So if it's going to share, it just needs one more, right? And then it's happy. But if you're chlorine and your valence electrons are 7, you also need, because you're trying to get to what? 8. Now, hydrogen has other options too, though, right? What else could hydrogen do? Give it away. It could give it away. And if hydrogen gives it away, it will be a what charge? Positive, and it'll be attracted to something that maybe it gave it away to that has a negative charge, like chlorine, right? And then you have HCl. Yes? Okay. All right. And you can tell when you look here, the little number is the atomic number because that's the number of protons. It makes sense that the bigger number is the atomic mass because it's protons plus neutrons. All right, good. All right, when you look at the periodic table, what determines an element's reactivity, how it behaves? What determines that? Number of valence electrons, because it's either trying to ditch, stay the same, or share. Yes? Ditch, steal, or share. Okay. And so when we look at the periods versus the groups all have the same number of what? Valence electrons. So they behave the same. Periods, you're at the same energy level. That's the horizontal on your period. All right? Next. We already did this. Covalent ionic bonds. You should be good. Polar, nonpolar. When you share fair, it is a nonpolar. Okay? Nonpolar if you share fair. If you share unfair and somebody has a tighter pull on the electrons, that is polar. That becomes really important when we talk about what important molecule? Water. It's an inorganic molecule, but important. What are the other two inorganic molecules that are important?
Oxygen and carbon.
Water, oxygen, and carbon dioxide. Those are your 3 important inorganics. What are your 4 important organics? Hydrates, proteins, lipids, nucleic acids. What are the 2 subs of lipids?
Fatty acids.
Fats and steroids. Okay, just checking. All right. So this becomes really important because in water, we are mostly made out of water, yes? And water has polar bonds between its oxygen, it's one oxygen, and its 2 hydrogens because oxygen has a higher electronegativity. It pulls on those electrons, yes? And so you have oxygens with partial negatives and hydrogens with partial positives. How this plays out is how one water molecule interacts with another water molecule And the way it arranges itself, its partial negatives attracted to a different molecule's partial positives. What kind of bond is that? Hydrogen bond. Okay. Weak, but collectively strong, right? Can the hydrogen bonds between water molecules kill you? Yeah. If you drop high, right, and you fall because of the cohesion, right, of the water molecules together. Yes? Remember that? Right? Okay. So hydrogen, guys, don't lose it. I have 10 more minutes with you, and don't we want to maximize our time together? Thank you. So hydrogen bonds are really important because our code book is DNA, and that's what holds the two strands of DNA together is those hydrogen bonds. Okay, that's another example of that. Plus, you're mostly made out of water. All right, so these are the properties of water because of these hydrogen bonds. High heat capacity because when heat comes in, that heat can be shared amongst all the hydrogen bonds. High heat of vaporization, because you are sharing it amongst all the hydrogen bonds, you have to have even more heat until it gains vaporization. Good solvent for anything that is hydrophilic, right? Hydrophilic means water loving. Things that are hydrophobic, it's not a good solvent for, like oils. Good. Cohesive, adhesive. Cohesive is water with itself. Adhesive is water with something else. Good. We see that when water goes up tall plants. High surface tension, we talked about that, is less dense when frozen. That's great because when water gets really cold and the ice is forming at the bottom of that lake super cold, it will what? Flow to the top, because as water freezes, the distances all become equal between the hydrogen bonds. So the space is bigger, so it's less dense, so it floats up to the top. That's distracting. Floats up to the top, and then it forms an insulating layer at the top so that the rest of the life below is still protected. And I think I gave you several slides expressing all of that. We can cool ourselves off. This is good solvent. So you can look through all of those. Don't have a seizure. Are we seizuring? Ah! Hang on. Let me see if I can break the seizure. Oh, okay. Sorry, hang on. Okay, you are not going to pause and tell me what you learned. I just had to do that. Pause. Tell me what you learned. All right. Guys, we have another old chapter, so can we please focus? Okay, so pH is a measure of what? Hydrogen ions. It's a negative log scale. Every change is a what? Tenfold. Yeah, 10 times change. So if we have a pH of 5, okay, and we drop to 3, from 5 to 4, it's a 10 times change. And then from 4 to 3, it's 10 times more concentrated. So 5 to 3 is 10 times 10. That's 100 times more concentrated, right? So it's a big deal each time you change in value. In our body, we have things to help us make. Now, oh, wait, let me back up. Enzymes. What are enzymes? Organic catalysts. What do enzymes do? Speed up the reaction by lowering the energy of activation. Enzymes work at a particular pH and temperature. So is this important? Yes, because something that digests food in your stomach requires a very what? Low pH, acidic, right? So we have to have ways to protect our bodies to create the right pH in the right location. Yes? Okay. Now, next on that, what helps us, like in our bloodstream, is buffers. Okay, buffers can help keep our pH just right. All right, that takes us now. Are you ready to chapter 3? Now, chapter 3 is all about carbon, right? Don't distract me. If you have to go, go, but we are working. So the way carbon works is we need to know that it has 4 valence electrons, right? Because it has a total of 6 electrons. Is that right? 2 in the first energy level, 4 in the next. That's great because it's going to tend to what? Ditch and steal or share? Share. So it can be hooked up with 4 other molecules, right, to build the complexity of your body. Yes? So that's why carbon's good. When you have a bunch of carbons and hydrogens together, they are called what? Hydrocarbons. On top of those hydrocarbons, you can attach important functional groups, like a hydroxyl group you could attach on there, an OH, or a carboxyl group. Do you reckon, or a carbonyl, sorry, here, carboxyl group, C double bond OH, where have we seen that? Amino acid. And look, here's an amino group. We can attach that onto a hydrocarbon. That's how we build all the different important organic molecules in our body. All right. We need to review both hydrophilic and hydrophobic, which we already know. Okay. Isomers. When I say there is a C5 compound, is every C5 compound the same? No, it's the way they're arranged. Yes? All right. And then this brings us here, the 4 important organic molecules. Carbohydrates are ultimately built out of, it's not hard. She says, carbohydrates are built out of what? You all know this. What are carbohydrates built out of? Sugars. Full stop. Come on, get it together. Don't celebrate your ignorance. Lipids. What are our 2 categories? Steroids are 4 carbon rings. Fats are a glycerol with 3 fatty acid chains. Those fatty acid chains could be or Good. If I remove one of those fatty acid chains and replace it with a phosphate group, phosphorus with oxygen around, I now have and phospholipids help build cell membranes, right? Proteins are ultimately built out of amino acids. Are proteins important? Yes, they are enzymes. They are channels, carriers, cell recognition, receptors, enzymatic junctions, right? So you know proteins are important. And then nucleic acids are built out of nucleotides. Nucleotides are composed of a base, sugar, and a phosphate group, right? What are the 2 or the 3 that we discussed as far as nucleic acids go? DNA, RNA, ATP. DNA and RNA are genetic, what starts with an I? Information, right? ATP is energy. Good. When I go back to proteins, proteins are built out of what again? Amino acids. When you talk about the sequence of amino acids in a protein, what is that called? The primary structure, right? But that primary structure, what comes after primary structure? Secondary structure. What are your 2 choices? Alpha. Now, how many different amino acids are there? 20 different amino acids, right? Now, is the primary and secondary structure dependent on what R group you have in your amino acid? No, it's because your amino groups and your carboxyl groups, right? What structure is dependent on the R groups? Tertiary structure. And that is the folding pattern imposed upon the secondary structure. And the hydrophobics will get together and the hydrophilics will get together, yes? Then what's the next level? Quaternary structure. And that's when you have more than one polypeptide, more than one chain. Now, how did we hook these organic molecules together? Let's take carbohydrates. Breathe just for a second. Carbohydrates are built out of what? Sugars. That's their monomer of sugar, right? I have 2 sugars. Are they happy by themselves? Yes, you know they are. So if I want them hooked together, I have to take something away so they become needy and hook up. Yes? So what is that reaction called? What is it? Dehydration synthesis. What's the other name? Not hydrolysis. Condensation synthesis. Come back. Hey, don't distract. We only have a little time. No drama. Dehydration synthesis or condensation synthesis. What do they have in common? Synthesis. It's about building, right? If I want to break them apart, what is it called? Hydrolysis. Hydrolysis. Water is lysine or separating. Does that make sense? So I have to interrupt that bond, and one of them gets an H and one of them gets a OH. All of this is not going to happen spontaneously. It's going to require a what? Enzyme. Do we know all about enzymes now? Yes, of course we do. Okay. Then what you'll find in here is where I just go through the bricks. Okay, I gave you the disaccharides here. I talked about polysaccharides. They are either for energy storage. Name your two energy storage ones. Glucose is a monomer. Starch in plants and glycogen in animals. Pause. Are there other polysaccharides than that? Yes. Are there other energy ones than that? Yes. I'm just giving you two. Structural molecules. Now give me a structural molecule. Cellulose in plant cell walls and chitin, which are in fungal cell walls or exoskeletons. All right. Good. Okay. Then, guys, I have literally 3 minutes with you. Okay. Lipids. Okay. You have fats and you have steroids. Fats, you have a glycerol molecule and 3 long fatty acid chains. These are not drawn to scale, right? They could be 16 to 18 carbons long, right? And those can either be, like we talked about, saturated or unsaturated. Okay. Then we talked about the other kind of lipid are steroids, right? Are steroids in cell membranes? Yes, they are. What is it? Cholesterol in animals, not plant cells, right? And it helps with the what of the cell membrane? Fluidity. Good. Are carbohydrates in cell membranes? Yes. What are they called? Glycolipids and glycoproteins. Okay, good. See? And then we talked a little bit about waxes. That's also a modification of fatty acid with a long chain alcohol. Proteins. We talked about all the many functions. No, baby. I didn't even touch. Hang on. Did it stop? Okay. Why does it? Yeah. Pause and write down what you know. Oh, she's still not pausing. Okay. Here we go. Okay. I'm going to try to keep going here. So then protein functions. Okay, this is the structure of amino acid. Remember, we have our amino group, our carboxyl group, our H, same, around a central carbon always. And then we have a what group? R group. Good. And how many different R groups? 20. What do we call the special bond between amino acids? Peptide bonds. It's still a dehydration synthesis though, right? Yep. Okay. Then we talked already about primary, secondary, tertiary, quaternary structure. Here is a nucleotide, base, sugar, phosphate. How many different bases do we have? 5, right? Remember we had purines and pyrimidines, right? Okay. Poor primitive pyrimidines, they only have 1 ring, whereas purines have 2. Adenine in DNA always hooks up with thymine, and guanine always hooks up with cytosine. Good. And then I just compared and contrasted DNA with RNA. And I gave you this summary in case that would be helpful for you. Remember that? And then that is, my friends, the end. Good job. Yay! There's like 4 or 5 of these. We have 5 of these.